Okay, so welcome back to this next video in which we are discussing Huntington's disease and Huntington's disease medications. Okay, right, so we've now looked at the physiological function of the basal ganglia in the motor system. We've looked at this cortico-basal ganglia thalamocortical pathway which controls the initiation of voluntary movements. Okay, now we are ready to talk about what happens in Huntington's disease, which portions of the basal ganglia degenerate. Now, the portion that degenerates is the dorsal striatum, okay? And this is what accounts for both the early motor symptoms that is seen, which is the chorea, and then later on the hypokinesia. Right. So in early Huntington's disease, the neurons which degenerate first are the indirect neurons. Okay, so the indirect pathway medium spiny neurons are the first neurons that are going to degenerate. Okay, now let's think about why this is going to lead to that early motor symptom of chorea. Now remember, what was chorea? It was involuntary jerky movements, okay, and also writhing movements, particularly of the neck. Generally what happens is you end up having these involuntary movements where you throw your head back, okay, you fully extend your head, okay, and expose your neck out a lot, okay, and that's a very sort of writhing movement. It looks as though you're writhing about, okay. You also have jerky movements of the hands and the arms and the feet and the legs as well. And remember the other thing, that these were particularly bad when you try to initiate a voluntary movement. So you do have chorea even when you're not trying to make voluntary movements, but particularly it's brought on when you try and initiate a voluntary movement. You then get loads of unwanted movements as well. Now, let's try and figure out why this makes sense in terms of the indirect medium spiny neurons degenerating. Okay, so remember, what was the purpose of the indirect pathway? The purpose of the indirect pathway was so that this portion of secondary motor cortex here, with the motor plan that actually needs to be initiated, it can make sure that permission is not going to be given to the other portions of secondary motor cortex which have junk motor plans which you don't want to be initiated. Okay, so remember, what will happen is this area will activate indirect pathway medium spiny neurons here, okay, and these indirect medium spiny neurons, which are in a particular portion of the dorsal striatum, will then inhibit a particular portion of the external globus pallidus. Okay, now that particular portion of the external globus pallidus, the neurons there were inhibiting a particular portion of the subthalamic nucleus. Now that these are no longer functioning, that portion of the subthalamic nucleus will now turn on, and it will activate many portions of the internal globus pallidus and substantia nigra pars reticulata. These will then inhibit many portions of the ventroanterior and ventrolateral thalamic nuclei, and these portions of the ventroanterior and ventrolateral thalamic nuclei that are being inhibited are the portions which will give permission to all the other portions of secondary motor cortex outside of the portion that actually has the motor plan that needs to be initiated. If we get rid of these neurons, if they've gone, then we destroy the entire indirect pathway. We destroy the removal of the permissive signal, or the sort of potentiation of the removal of the permissive signal. Because remember, you know, the internal globus pallidus and the substantia nigra pars reticulata are inhibiting these areas all the time. We're just upregulating that inhibition. Okay, so if we remove the upregulation of that inhibition by destroying the indirect pathway, then whenever you initiate a voluntary movement, you also end up getting this chorea. You get unwanted involuntary movements, and that can be explained by this, because now we're no longer upregulating that inhibition of the permission signal. So now it seems that some motor plans are slipping through and being sent from the secondary motor cortex to the primary motor cortex that should not have been sent through. Okay, and hence that's how we can explain the chorea in the early stages of Huntington's disease. And remember, this chorea was called Huntington's chorea, and it really is the cardinal symptom of Huntington's disease. Okay, then, now let's move on to the later stages of Huntington's disease. Okay, so in the late stages of Huntington's disease, the direct pathway medium spiny neurons are going to degenerate. So the first neurons that go are the indirect medium spiny neurons, and then in the later stages of the disease, the direct medium spiny neurons go. Okay, now let's go back to the direct pathway. This is going to explain why in the later stages of Huntington's disease you stop having the hyperkinesia and instead you develop hypokinesia. You lose the ability to initiate voluntary movements at all, really, and you just become rigid and still. Okay, right. So now the direct pathway medium spiny neurons, this one here and this one here, those are going. Now let's just remind ourselves of the direct pathway. Remember, if this portion of secondary motor cortex, which has got the motor plan that actually needs to be initiated, wants the motor plan to actually be initiated, it has to send activatory signals to this area of the dorsal striatum, and it's going to activate direct pathway medium spiny neurons in that area of the dorsal striatum. These will go into certain areas of the internal globus pallidus and certain areas of the substantia nigra pars reticulata and inhibit the neurons in those areas. Hence, these neurons will now stop functioning, which means that they will no longer be inhibiting a certain portion of the ventroanterior and ventrolateral thalamic nuclei, and that specific area will now send permissive signals back to this specific area of the secondary motor cortex, and that gives the permission for this motor plan to now go on to the primary motor cortex. If we lose these direct pathway medium spiny neurons, these are now gone, okay, we're going to lose the direct pathway. We're going to lose the ability to initiate voluntary movements at all, and that's why you get hypokinesia. Okay, so in the early stages of Huntington's disease, you lose the indirect pathway. In the later stages of Huntington's disease, you lose the direct pathway, and this causes the early motor symptoms of Huntington's chorea, and then the later motor symptoms of hypokinesia. Okay, right. So that then now explains the motor symptoms then of Huntington's disease in terms of the degeneration of the dorsal striatum. Okay, we're now going to discuss medications, okay, used to treat Huntington's disease. Now, none of these medications are going to stop the neurodegenerative process. All of these medications are going to simply work to reduce the chorea. They are really treatments for Huntington's chorea. We can stop you having the involuntary movements, basically. Okay, so there are two types of medications that I'm going to talk about here. Okay, the first type are dopamine depleting agents. Okay, and then the second type are antipsychotics. Okay, and both of these types of drugs are going to work on those dopaminergic neurons, or rather the dopaminergic neurotransmission that occurs within the dorsal striatum, which is occurring because of those neurons from the substantia nigra pars compacta. So they're both going to work on the nigrostriatal tract, basically. Okay, so also antipsychotics. Okay, so let me give you the idea, basically, behind how these drugs are going to work to treat the chorea, okay, and then I'll explain in more detail how each of these drugs works. So the basic idea is this. Remember, the substantia nigra pars compacta here is sending dopaminergic neurons into the dorsal striatum, okay, and these are chronically and tonically releasing dopamine onto the medium spiny neurons of both the direct and indirect pathways of the dorsal striatum. Okay, so now remember, in the early phases of Huntington's disease, where you've got Huntington's chorea, it is the indirect pathway medium spiny neurons that have degenerated. So let's ignore the direct pathway ones. Let's assume that's working fine. Okay, it's the indirect pathway neurons that are now under functioning. Okay, so how can we up the function of the indirect pathway medium spiny neurons? Okay, you've not lost all of them yet. You've lost quite a few of them, but we want to now upregulate the activity of the ones that are remaining. Now, how can we do that? Well, remember what dopamine was doing. dopamine decreases the activity of the indirect pathway. Dopamine acts on D2 receptors, which are on the necks of the dendritic spines of indirect pathway medium spiny neurons, and desensitizes them to glutamate. Okay, it reduces the amount of sodium current that a burst of glutamate is going to let into the cell, and thereby reduces the chance that this indirect pathway medium spiny neuron is going to fire. So if we were to remove the effect of dopamine, okay, if we were to stop dopamine acting on the remaining indirect pathway medium spiny neurons, then we could make the remaining indirect pathway medium spiny neurons more excitable, okay, and therefore we would up the amount of indirect pathway activity, basically, and that might compensate for the loss of indirect neurons that you've suffered because of the Huntington's disease. Okay, so this is the idea, stop the dopaminergic transmission here. Now, dopamine depleting agents, these ones here, are going to work by stopping the release of dopamine from the axon terminals here, whereas the antipsychotics are going to stop the dopamine acting on the D2 receptors on the medium spiny neurons. Okay, so let's now discuss this in a little bit more detail. Let's start with the dopamine depleting agents. Okay, now to explain the dopamine depleting agents, we need to have a look at a dopamine axon terminal in a little bit more detail. Okay, so let's say this is the axon terminal of a dopaminergic neuron that has come from the substantia nigra pars compacta. Okay, right, so we need to discuss how is dopamine packaged into synaptic vesicles, because that's where the dopamine depleting agents are going to interfere, basically. So we'll start off with the synthesis of dopamine in the axon terminal. Now, I'm not going to actually go through the molecular process. All I'm going to say is that dopamine is synthesized from the amino acid tyrosine in the axon terminal. Okay, we don't need to know anything more than that to understand this. So Y is the single letter code for the amino acid tyrosine. Okay, DA here is an abbreviation for dopamine that I'm going to use a lot here. Okay, right, so let's add a bit of colour onto this to make it look a little bit more interesting. So here is dopamine, and here is tyrosine in red here. Okay, right. Now, once you've synthesized dopamine in the axon terminal, the dopamine is not just going to be released now, okay, you're only going to release the dopamine once an action potential arrives. So first what's going to happen is the dopamine is going to get stored in a synaptic vesicle here, okay, and then when an action potential arrives, these synaptic vesicles will fuse with the presynaptic membrane. Okay, so I'll kind of show this here. What will happen is this membrane here will fuse with this membrane here will fuse with this membrane, and then you'll open up a pathway from the lumen of the synaptic vesicle out into the extracellular fluid, and that's how you'll release the dopamine. Okay, so this is a synaptic vesicle here. Okay, so the dopamine is going to be stored in synaptic vesicles until an action potential arrives. Now, how do we get it into synaptic vesicles? Well, we use secondary active transport, but secondary active transport relies on an ion gradient, okay? And this ion gradient is going to be a proton gradient across the membrane of the synaptic vesicle. And I firstly want to explain where that proton gradient comes from. Okay, so basically there's going to be a much higher concentration of protons in the lumen of the synaptic vesicle than there is in the cytoplasm. And the reason for this is that there is a primary active transporter on the membrane of the synaptic vesicle, which is known as the vesicular ATPase. Okay, so this stands for the vesicular ATPase, the VATPase for short. Okay, so I'll colour in this VATPase here in turquoise. Now this is going to be an example of a primary active transporter, okay, because it is going to transport protons into the lumen of the synaptic vesicle by getting energy from a chemical reaction, okay, so the energy that it needs to move a proton into the lumen of the synaptic vesicle is going to come from a chemical reaction, and that chemical reaction is going to be the hydrolysis of ATP, hence why this is called an ATPase. It's going to break down ATP. Okay, so ATP is going to be hydrolyzed to ADP and inorganic phosphate, and the energy released by this hydrolysis is going to be used to move one proton from the cytoplasm of the axon terminal into the lumen of the synaptic vesicle. Okay, so you build up an ion gradient using this V80Pase, and now you're going to have a secondary active transporter which is going to use this proton gradient to move dopamine into the synaptic vesicle. Okay, so here in pink, this is what is known as VMAT2. Okay, now VMAT2, I'm not going to put it there because I want to write secondary active transporter there in a moment. In fact, no, I will put it there, I'll write secondary active transport somewhere else. Okay, sorry for that indecision. So VMAT2 stands for the vesicular monoamine. Okay, so V is for vesicular, MA is for monoamine, M for mono, A for amine, and then transporter 2. Now there is a VMAT1, there is a vesicular monoamine transporter 1, but it's not generally used in neurons. It's used in cells, for instance, the cells of the adrenal medulla, which make adrenaline and have to store it in vesicles as well. Okay, and in axon terminals you generally use VMAT2. Okay, now VMAT2 is going to be an example of a secondary active transporter. Okay, now what this means is that, again, you're going to be moving something up its electrochemical gradient. Okay, so it has to be an active transport process. Okay, however, you're not going to get the energy needed to move the dopamine into the synaptic vesicle from a chemical process. Instead, what you're going to do is you're going to use an ion gradient that already exists. So what you're going to do is you're going to use this proton gradient. You are going to allow two protons to leave the synaptic vesicle lumen and go into the cytoplasm. So two protons are going to leave, and you're going to use the energy that's released by those two protons going down their electrochemical gradient. You're going to use that energy to move the dopamine up its electrochemical gradient into the synaptic vesicle, and that's how you're going to get the dopamine in. So one dopamine goes in for two protons out, basically. Okay, and that's how you store the dopamine in the synaptic vesicles. Now, not all of the dopamine you produce ends up actually in the synaptic vesicles. Some of it will be broken down. Now, the main enzyme that breaks down dopamine in the cytoplasm of the synaptic terminal here is an enzyme known as monoamine oxidase. Okay, and for short, monoamine oxidase enzymes are often abbreviated as MA for monoamine and then O for oxidase. Now there are two forms of monoamine oxidase A and monoamine oxidase B. Both of them can break down dopamine, however monoamine oxidase B is much better at it than monoamine oxidase A. Okay, so this is the main one that's going to break down dopamine. Now, where are these enzymes located? Because they actually have a pretty interesting location. They're located attached to the outer membrane of mitochondria. So let me draw a mitochondria for you here, and then I'll show you where they are going to be located. Okay, so remember, mitochondria have a very interesting membrane structure. Okay, they have two membranes. An inner membrane, which is the more internal one, okay, here, which is invaginated inwards in these structures known as cristae, okay, and that's to increase the surface area of the inner membrane, because remember, the inner membrane is the site of oxidative phosphorylation, it's the site of the electron transport chain, okay, it's where you make ATP from reduced NAD and reduced FAD. Okay, then the other membrane is the outer membrane, okay, and then in between the two of them you have an intermembrane space. Okay, now, monoamine oxidase B is actually going to be attached to the outer membrane of the mitochondria. So if I now draw the outer membrane in more detail, so now I'm not drawing the outer membrane anymore as a single line, I'm drawing it as two lines, and this potentially is extremely confusing here. Okay, these two lines that I have drawn here are no longer representing the outer and inner membrane. This is just the outer membrane. I'm drawing the two layers of the outer membrane. Remember, membranes, biological membranes, are phospholipid bilayers. So this is just the bilayer of a single membrane. Okay, the outer membrane. I haven't even shown the inner membrane here. If you like, I'll put on the inner membrane if that makes it clearer. Okay, here's the inner membrane with the intermembrane space in between them. Okay, right. Now, monoamine oxidase B then is going to have its amino terminus in the cytoplasm. Okay, so up here. So if I draw it in the smaller picture as well. Okay, here's the amino terminus, like so. Then you're going to have the main enzymatic part here, also in the cytoplasm. Then you'll have a single membrane spanning alpha helix, which spans the outer membrane, and then the C-terminus will be in the intermembrane space here. Okay, so let's colour in monoamine oxidase B in orange here. Okay, there we go. And this is also it down here. Okay, so that's monoamine oxidase B here. And this is capable of breaking down dopamine. Okay, so either the dopamine that you produce is going to get pumped into this synaptic vesicle, or it's going to be destroyed by monoamine oxidase B, which I'll now show here. Okay, so here is our mitochondrion here. Okay, and then we'll have our monoamine oxidase B here. Okay, so either the dopamine can get stored into the synaptic vesicles and actually get released in the dopaminergic transmission, okay, or it can be broken down by our monoamine oxidase B here. Right, so that's all that we're going to need to understand about dopamine axon terminals to now understand the dopamine depleting agents. Okay, so let me give you the two examples of dopamine depleting agents. So there is reserpine, okay, which is an old drug that isn't really used that much anymore, but it can be used for Huntington's disease. It has a number of inconvenient side effects, basically. Okay, the other dopamine depleting agent also has a number of side effects, but it's not quite as side effect rich as reserpine. Okay, so the more common drug that's actually used is tetrabenazine. Okay, so here it is, tetrabenazine, but reserpine is also a dopamine depleting agent. So both of these are dopamine depleting agents. Okay, right, now what do? Basically, both of these drugs bind to the vesicular monoamine transporter 2, VMAT2, and block it from functioning. Okay, so they are going to bind to VMAT2 and block it here. Okay, and hence, once you've blocked that from functioning, dopamine is no longer going to get transported into synaptic vesicles. Okay, instead it's all going to get destroyed by monoamine oxidase B. Your synaptic vesicles are going to end up depleted of dopamine. Okay, hence why they're called dopamine depleting agents. And now those dopaminergic neurons synapsing onto the necks of the dendritic spines on the indirect neurons, okay, and also the direct neurons, but let's not worry about that, okay, those dopamine fibers that are synapsing onto the necks of the indirect neurons here are now not going to be releasing dopamine, and therefore the dopamine is no longer decreasing the sensitivity of the indirect medium spiny neuron here. Okay, so now the sodium currents induced by a glutamate burst are going to be bigger, and therefore these indirect pathway medium spiny neurons are going to fire more often, and hopefully that will make up for the lost medium spiny neurons of the indirect pathway that you've had. Okay, right. So that's how the dopamine depleting agents are hopefully going to stop the choreiform movements, okay, the chorea of early Huntington's disease. The other drugs that you can use are antipsychotics, which are going to block the D2 receptors. Okay, so remember, on the necks of these dendritic spines, you have D2 receptors on the medium spiny neurons of the indirect pathway, in a way. It's D1 on the medium spiny neurons of the direct pathway. Okay, if you could put a drug that would bind to the D2 receptors and not activate them, but would stop dopamine from being able to bind to them, okay, that would stop the dopamine from actually functioning at those dopaminergic synapses onto the indirect pathway medium spiny neurons. So let's draw this. Okay, so let's say this is the membrane of our indirect pathway medium spiny neuron. Okay, our indirect MSN, I'll call it. Okay, then we've got our D2 receptor here. Okay, and we know that this D2 receptor, when activated, will cause desensitization of that dendritic spine, i.e. it will mean that the sodium current that you're going to let in for a glutamate burst released by the cortical glutamatergic neuron is going to be smaller. Okay, so the indirect pathway medium spiny neuron is therefore less likely to to actually fire an action potential when dopamine is acting on these D2 receptors. So if we could put a drug molecule here that would bind to the D2 receptor where the dopamine wants to bind, okay, but does not activate the D2 receptor, then that would block the dopamine from being able to actually activate the D2 receptor, and therefore it would block the effect of dopamine on the indirect pathway. Now dopamine can't actually bind, okay, so it will go off. Okay, this sort of a drug which binds to the D2 receptor but does not stimulate it is known as a D2 antagonist, okay, which you should contrast to an agonist. An agonist is a molecule which binds to a receptor and activates it. An antagonist is just a molecule that binds to the receptor and does not activate it, and blocks the dopamine from actually binding there. Okay, so D2 antagonists are used hugely as antipsychotics. Okay, they stop psychosis, which is another name for schizophrenia, okay, which involves hallucinations and delusions. Okay, so antipsychotics are all D2 antagonists, and that's believed to be crucial to how they work. Okay, but we can now make use of them here to treat the chorea in Huntington's disease. Okay, so let me give you some examples then of antipsychotics. Okay, so I'll start off with the ones that are particularly used in Huntington's chorea. Okay, so some that are particularly used in Huntington's chorea are pimozide, okay, and also sulpiride, okay, in addition to haloperidol. Okay, and then I'll give you some more antipsychotics that aren't as commonly used for Huntington's disease, but they could be used for Huntington's disease. So other examples of antipsychotics that can be used are chlorpromazine, okay, and also risperidone. And these are all D2 antagonists. Oops, spelled risperidone wrong. Risperidone. Okay, so particularly the first three are used in Huntington's chorea: pimozide, sulpiride, and haloperidol, but chlorpromazine and risperidone can also be used. Okay, right, and again they will increase the activity of the indirect pathway is the idea, and therefore hopefully block these unwanted movements that occur whenever you try to initiate a voluntary movement because of the underactivity of the indirect pathway that has occurred because of the degeneration of the indirect pathway medium spiny neurons in the dorsal striatum in early Huntington's disease. Okay, so that now concludes our discussion of Huntington's disease and Huntington's disease medications.